Thank you very much all for coming, and thank you organizing committee for giving me the opportunity to present the work on behalf of my colleagues. The first presentation is going to talk about use of non-specific immune stimulant based on mycobacterial cell wall fraction and its ability to attract neutrophils in the lumen of uterus in dairy cows. So when we talk about reproductive performance in dairy cattle, we usually think about the measure of the speed, how quickly the cow can get pregnant after the voluntary waiting period. And the parameters that we measure are usually days to the first service, calving interval, conception rates, and so on. And those are all parameters very important because they relate to economic benefit and profitability of the dairy farms. Factors that may affect the reproductive performance include, but they're not limited to, negative energy balance, resumption of ovarian cyclicity, postpartum diseases, mostly when we talk about metritis and endometritis, and infectious-related diseases that will lead to abortions that are usually caused by leptospirosis, IBR, BVD, or mycoplasma. Most of the time when we talk about postpartum diseases, we refer to the puerperal bacterial contamination of the uterus, and the factors that risk factors that are associated with that are the stress due to high milk production and housing issues because of the overpopulation and exposure to potential pathogenic strains and ease of distribution. Some of the consequences that relate to the reduced reproductive performance relates to the fact that the cow can become pregnant or increase the calving interval. Some of the common treatment options include antibiotics and hormonal therapy. So what we decided to do is to target some of those conditions, primarily endometritis and metritis, by using non-specific immune stimulant that is based on mycobacterial cell wall fraction derived from the non-pathogenic Mycobacterium flea, which is the gram-positive soil-borne bacteria. The composition of mycobacterial cell wall fraction that we manufacture contains the DNA and RNA that is associated to and attached to the cell wall components as well. Currently, the product or this formulation is licensed in US and Canada, and right now we're waiting for the approval in Australia and New Zealand. When we look at the chemical structure of the formulation, the two major components that are highlighting this formulation are mycolic acid and moramyl dipeptide, which are considered as a major antigenic and immunodominant components. And it is very well known that these two parameters can significantly induce the immune response. So when you talk about the mode of action that is induced by MCWF, we can say that as a non-specific immune stimulant, it has the ability to induce innate and adaptive immune response. When I talk about innate immunity, we usually refer to the high influx of the neutrophils as well as some NK and NKT cells. On the other hand, it can also stimulate the cascade of reactions that will lead to the production of different cytokines and activation of the macrophages and dendritic cells. So overall, we can say that the product or formulation can induce the humoral and cell-mediated immune responses. Based on the variety of the studies that we performed in the past, in vivo and in tissue, in vivo and in vitro, and using different animal models, some overall mode of action would relate to activation of the immune system receptors, such as the TLR2, NOD2, cytokine production by monocytes, macrophages, dendritic cells. And again, we have the variety of the cytokines that can be connected to either Th1 or Th2 immune responses. In addition to that, we also observed that the product can induce the GSG and CEF product. So again, the overall induction of the cell-mediated and innate immune responses observed. So lately in the bovine industry, not only bovine industry, overall in the veterinary industry, it's a high trend of using the immunotherapeutics for a variety of indications. The immunotherapy has a very high presence in veterinary oncology, and lately it's been shifting in the bovine field as well. When we talk about the bovine potential immunotherapeutic indications, we can say that would be very applicable to be used in the to control the calf health, primarily diarrheas, pneumonia, pneumonia shipping, metritis, clinical mastitis, and could be used in the dry cow therapy or for the treatments of the respiratory diseases. All these indications are again associated with the high use or abuse of antibiotics. So we think that the use or finding the appropriate alternatives for the use of antibiotics would be beneficial to veterinarians and producers as well. So the main objective of the present study was to evaluate the efficacy of 3 different doses of mycobacterial cell wall fraction to induce the neutrophil influx following the intrauterine administration. And we wanted further down to evaluate the functional activity of those polymorphonuclears by measuring the reactive oxidative species that are released by those attracted neutrophils. The rationale for that is that we believe that the high influx of the neutrophils can be achieved with the different concentrations in dose-dependent manner following intrauterine administration. So the study design included enrollment of the 40 lactating cows, roughly around 45 days in milk, with an average history of easy calving and no retained placenta. On the clinical examination, we established the absence of the clinical signs of the clinical endometritis and an average body condition of 3. In addition to that, we looked at cytological examination, and the cutoff value for the determining the endometritis was less than 8% of the polymorphonuclears per 300 cells. For the purpose of cytology examination, what we did as well, we designed a new catheter or swab that is slightly different from the cytobrush in terms that is completely made of the plastic, and instead of the cytobrush, the upper tip is made from the special plastic silicone base that has very high adhesive power. So basically, by using this catheter, we avoid damaging the endometrium, but still having the significant number of cells that were attracted and collected, and giving us nice smears down the road. We also compared this catheter to the cytobrush before to make sure that there is a correlation and the consistency. And the manuscript within preparation, we're going to show that we are as good or about 15 to 20% more accurate than the cytobrush alone. So with the study design, like I said, included 4 experimental groups with 10 animals per group. Animals in group 1 received oyster glycogen as a positive control. Animals in group 1 received 2.5 ml of mycobacterial cell wall, which correlated to roughly 0.6 milligrams. Animals in group 2 received 5 ml, correlated to 1.25, and animals in group 3 received 10 ml of mycobacterial cell wall fraction, correlating to 2.5 milligrams of the formulation. Mycobacterial cell wall was diluted into physiologic cell line up to 60 ml, and infused into uterus equally into each horn, roughly 30 mils into each horn. At 24 hours post-administration, we performed another cytology sample and uterine lavage, collecting the cells and subjecting them to the phago burst flow cytometry analysis to define the oxidative burst index. So the next few slides will show the cytology samples, like at the zero time point and 24 hours after, with a significant influx of the neutrophils in each of the experimental groups that we tested. Looking at the graph, we were able to observe statistically significant influx of neutrophils in each of the experimental groups compared to their baseline values. In average, like it was more than 50% increase of the neutrophils in the lumen of uterus, either by using the oyster glycogen or 10 ml MCWF, while with the 2.5 ml we had roughly 15% or 45% neutrophil infiltration in the lumen of uterus. The next step was to assess and measure the percentage of those attracted neutrophils for their ability to release the reactive oxidative species, and this was measured again by using the commercially available kit PhagoBurst. And like you see, there was no statistical significant differences, but there was a numerical difference between the groups that you can see that there was a dose correlation between the 3 experimental groups that received our product compared to the control. And for those who are less familiar with the reactive oxidative species, for the neutrophils to function properly and to kill the pathogen, they're required either for the intracellular killing or they can be released extracellularly and enable, again, proper neutralization of the pathogen. So just a brief overview how we measure. So the upper left image shows the negative control where there was no reaction. The upper right one shows the positive control where you can see how the cells shifted with the reaction of the mean fluorescent intensity. And the upper lower one, the lower left one shows the weak reaction, while the lower right one shows the very strong reaction to the mean fluorescent intensity. The next one shows the actual values of the mean fluorescent intensity for the neutrophils that were positive for ROS activity. And again, we were able to see dose-dependent correlation between their mean fluorescent intensity and the number of neutrophils that were, and the concentration of the product that was used in the study. And lastly, we wanted to see what would be the oxidative burst index, which is basically a mathematical correlation between the percentage of ROS-positive cells and the mean fluorescent intensity divided by 100. And these graphs actually showed us clear dose-dependent response when compared to the concentration of the antigen that was used, meaning that the 5 ml dose showed the most consistent and most the strongest reaction between the 3 doses that was used. What was even more interesting is that even though at the beginning, if you remember the slide that I showed the neutrophil influx, even though they were like in a similar levels, however, here we can see that those neutrophils attracted by the presence of the mycobacterial cell wall fraction were stronger in terms of their oxidative potential, oxidative burst potential, meaning that they're more ready to attack and phagocyte the pathogens if they're present. So with this in mind, we further followed those animals and measured their reproductive performance as well. There was no any differences in days in milk. Service period was around varied from 93 for the 2.5 ml to up to 150 for the 10 ml dose, while controls had almost 170 days. What was also interesting is to see that the animals that received our product had almost 1 time less artificial inseminations compared to the controls, and conception rates were pretty much high. Again, this is just proof of concept and pilot results, but it was good indication that we have to move forward in a clinical setting using more animals to justify its use and its potential for the treatment on endometritis and improving the reproductive performance. So overall, the conclusion would be to say that the MCWF has a potential to attract significant numbers of polymorphonuclears following single intrauterine administration, that those PMNs that are attractive have the strong oxidative burst activity and could lead to accelerated clearance in case of the postpartum infection and establishing the uterine homeostasis. The dose of 5 ml seems that the most appropriate based on the reaction in terms of how strongly and those neutrophils that were attracted can react and attack the pathogen. And we performed additional studies looking at the different routes of administrations using the same 5 ml and giving the formulation in the intravulva, IV, and subcutaneously, and again measuring the cytology, complete hematology and biochemistry, and reproductive performance as well. So with this, I would like to thank you and leave a few minutes for any questions that if you have.
Yeah,